This is the distinguished public lecture by Professor Sarah Teichmann. And before we get started, I request all of you to keep your mobile phones in the silent mode. And I remind you that this talk is being live streamed and so that it's accessible to a much wider set of fellows and associates of the Academy elsewhere in India, as well as general public from all over the country. So, and we now get started to introduce our speaker, Professor Teichmann. I request the past president of the Indian Academy of Sciences, Professor Partha Majumdar, to do the honor.
Good morning, everyone. In the morning, the president of the Academy introduced the Raman Chair Professorship to those of us who were present here. I know that many people have arrived just now for her talk. So I will just say one or two sentences about the Raman Chair, which was instituted in 1972 by the government of India to commemorate the memory of the founder of the Academy, Sir C.V. Raman. As the president mentioned, this chair is usually offered to very eminent scientists from around the globe, and this year we have offered this chair to two people, to occupy this chair to two very eminent scientists, one of whom is here, Professor Sarah Teichmann, and the other person will give a talk sometime later. Her name is Aviv Regev. She is currently with Genentech, but she was up until recently with the Broad Institute of MIT and Harvard. The last two occupants of the Raman Chair are also notable. Of course, they're all notable, but the last two occupants of the Raman Chair were Professor Harold Varmus, Nobel laureate, and the eminent physicist from Harvard, Professor Suvir Sachdeva. So as I said that this year we have two people occupying the chair. And although I actually have not gone through the record systematically, I'm pretty confident that ever since it was this chair was instituted by the Academy through the funding of the Government of India, this year we have the two youngest people occupying the chair. I'm pretty confident about that, but I cannot be certain because I actually haven't done my homework very systematically. So let me now introduce today's speaker, Professor Sarah Teichmann. She did her PhD at the MRC Laboratory of Molecular Biology in Cambridge, did a postdoc at the University College London with a Bate Memorial Fellowship, which is one of the most prestigious and competitive fellowships for postdoctoral research. She established her own research group at the MRC lab in 2001, where her main discoveries included the finding that protein assembly pathways are stereotypical and conserved. In 2013, she transitioned to the Wellcome Genome campus, where she became the first and to date the only faculty member appointed across both the EMBL European Bioinformatics Institute and the Wellcome Sanger Institute. Very prestigious position. In 2016, she was appointed as head of the cellular genetics program at the Wellcome Sanger Institute, and she co-founded, along with Aviv Regev, the Human Cell Atlas Initiative, a global consortium. Earlier, she's also been the director of research at the Cavendish Laboratory in the physics department of Cambridge University. From April 2024, she was appointed chair in stem cell medicine at the University of Cambridge. Additionally, Sarah dedicates a part of her life, part of her time, to GlaxoSmithKline and to EnsoCell Therapeutics, the startup company that she co-founded. Her lab focuses on developing and applying cell atlas technologies to understand human tissue architecture, particularly examining how cellular diversity is generated in the immune system during development. Sarah Teichmann and Aviv Regev are the founders of the international consortium, the Human Cell Atlas, with the mission to create comprehensive reference maps of all human cells as a basis for both understanding human health and diagnosing, monitoring, and treating disease. Sarah has received numerous awards and honors. I will only mention a few. She's received the Fellowship of the Royal Society in 2020, Fellowship of the International Society of Computational Biology in 2016, and Fellowship of the Academy of Medical Sciences in 2015. She's received the GlaxoSmithKline Award of the Biochemical Society of the UK in 2020, Mary Lyons Award of the Genetic Society UK in 2018, and the EMBO Gold Medal in 2015. I'll not provide you with a complete list of her decorations. She's very well decorated. She's a fantastic speaker, and of course, a very, very eminent scientist who has set the ball rolling for us to be able to analyze cells one at a time. Sarah.
Thank you so much, Partha, and also Umesh, for having me here. And thank you, everybody, for coming this morning. It's a tremendous honor and a pleasure to be speaking, and very, very excited about this Raman Chair. So, as Partha said, with the Human Cell Atlas Consortium, which we founded in 2016, our mission is to create a comprehensive reference map of the types and properties of all human cells, the fundamental units of life, as a basis for understanding, diagnosing, monitoring, and treating health and disease. And this morning, in the previous session, basically, we discussed kind of the history of this project, that includes scientists from across the globe, including here in India. And in this talk, I'm going to focus on basically some stories about the applications of the Human Cell Atlas. And I'm going to start with the pandemic, because the Human Cell Atlas has been used to tackle that unseen challenge, which kind of surprised us all, I think. And it's providing foundational knowledge that can help us address human health challenges. And the arrival of COVID-19 in coming across from China in early 2020 posed the key question of how exactly does the SARS-CoV-2 virus enter the human body? And of course, we knew that the viral entry factors were ACE2 and TMPRSS2, and that those are the genes that are crucial for the virus to dock and then enter the cells. And so any cells that expressing those two factors would have a sort of doorway for the virus to enter. And so what we decided to do was to harness the Human Cell Atlas data to ask which cells express those genes that are needed for the virus to enter the body, and which cells kind of have those patterns. And at that moment, in this terrible sort of public health emergency, the Human Cell Atlas community really banded together in an incredibly fast and an effective way to answer this question by pooling all of our data sets, regardless of whether they were published or not. So it was really a moment of spectacular kind of open global collaboration, and then analyzed the data. And so we collected and then computationally integrated data from approximately 50 million cells or so, and that revealed that the viral entry factors show multiple potential routes for entry, and then also for dissemination within the body. And all of the, or some of the tissues that we found had these entry factors are shown here. And in particular, we confirmed that there are cells in the eyes, the nose, and the mouth that were important routes for disease transmission and spread. And in the eyes, this was unpublished data that was shared by Linda Lako from Manchester at that time, and literally sort of when the shutdown was happening, she basically sent the data sort of across to us, and we basically integrated the corneal and conjunctival data and saw that there were specific epithelial cells in the conjunctiva that had the viral entry factors, secretory and ciliated cells in the nasal epithelium, ductal epithelial cells and salivary glands in the oral cavity. And of course, all of those cells are critical basically for horizontal transmission of the virus. And in particular, we later worked with Kevin Bird and others in the Human Cell Atlas craniofacial network that showed that the in the salivary glands, basically in those ductal epithelial cells, the cells are actually shed into the saliva and are in aerosol. They're aerosolized basically during speech, sneezing, singing, and so on. And so those findings fed into policy debates about mask wearing and disease transmission. In the UK, we had the SAGE committee of the government. And within the airways, the Human Cell Atlas data showed that the viral entry factors are more highly expressed in those nasal epithelial cells, so the secretory and the ciliated, rather than the alveolar epithelial cells, the AT2 cells in the alveoli deep down in the lung. And that sort of explained why, for instance, Omicron, which has a higher affinity kind of spike protein, is actually less damaging than, for instance, the Delta variant that goes deep inside the lung. So what this illustrates is basically that even the healthy reference data, so without having any disease data, the healthy reference data of the molecular content of the individual cells in our body, so this high resolution, deep molecular breadth data of the healthy tissues serves as a guidebook sort of about where viral entry factors are expressed, for instance, and will be extremely valuable for many different purposes. So by building this knowledge of the healthy reference cells and tissues in our body, we can ask lots of different questions. So what are the types of questions that we can ask? Examples are, for instance, where are those viral entry factors located, not only for COVID, but for other pathogens? So where are other pathogens entering? How are they disseminating inside the internal tissues? Where are genes expressed that are involved in disease, either rare disease variants, for instance, or common complex disease risk variants? Where are hormones expressed? And where are hormonal receptors in the body? So how is the endocrine system basically coordinating our physiology? Where are drug targets expressed? So where are drugs acting in a highly precise way, both in terms of their intended kind of cellular targets and also in terms of unintended targets that cause side effects? Where are surface molecules on each cell type that can act as a GPS system for drugs or lipid nanoparticles to be addressed to specific cell types in a highly precise way? So the data coming out of the Human Cell Atlas allows us to learn lots of different things about the healthy kind of mechanisms of action of our physiology, but also information that's useful for disease and for drug discovery. So this guidebook principle is something that I'd like you to think about kind of in terms of the application and utility of the Human Cell Atlas data. So I'm going to start off with a second example from application of Human Cell Atlas to SARS-CoV-2, because of course the pandemic basically had a big sort of impact in terms of guiding kind of our research interests. And so in particular, this is a very recent study that was published only a couple of weeks ago, but we actually started it quite early in the pandemic. And this is the UK SARS-CoV-2 infection challenge. And this was basically started in late 2020, early 2021, where the Chris Chu at Imperial College and colleagues got ethical approval from the UK Vaccine Task Force and support from the UK Vaccine Task Force to infect 36 healthy young volunteers between the ages of 20 to 30 that had not been vaccinated and had not been infected with the virus, so they're seronegative, to be intranasally inoculated with the pre-Delta strain of the SARS-CoV-2 virus. And so why is this study so exciting and so important? The reason that it's so exciting is that usually when we study disease samples from COVID-19, we get them from people who have symptoms. Okay, so they're people who already have a fever, cold-like symptoms, pain, etc. But at that point, the virus has already been inside their body for 3 or 5 days. Whereas with this infection challenge of these healthy volunteers, we're able to study the very early cellular responses that are actually pre-symptomatic and what we used was nasal swabs and blood studied by single-cell sequencing. And so this allowed us to answer questions such as, why do some people develop COVID-19 while others don't? Because there are some people in this cohort, even in this small cohort, that actually didn't develop any symptoms whatsoever, and some people who even managed to defend themselves from any viral entry into their cells. So those are people that we would never get to see in the standard kind of studies of COVID-19, where we're only getting samples from people with clinical symptoms. So that's a very special and very exciting opportunity to allow us to also identify potential mechanisms that confer protection. So single-cell transcriptomics can provide us with deep insights into the cellular responses, and having this longitudinal sampling basically gives us an opportunity to study this scenario where participants are intentionally exposed to the virus, of course, within a highly safe environment in a hospital setting. And so we had funding. Basically, this was all started during the pandemic, where it was very difficult because we had grants and funding for our non-SARS-CoV-2 studies, and had to very quickly get support basically for these SARS-CoV-2 studies. So this was, I basically felt this was a golden opportunity, and also called contacts basically in 10x Genomics and Illumina and so on, who basically enabled this study with reagents that were free or at very low cost because of this emergency situations, very precious samples. And so out of the 36 volunteers that were infected, we had enough funding to study 16 healthy young adult seronegative volunteers. And even amongst those 16, you can see that there were 6, so those are the red little people up there, who suffered sustained infection. So those 6 people had multiple positive tests, so there was PCR and antigen testing, and cold-like symptoms. There were 3 people that had only transient infection, so they had only one single positive test, but then negative, so only very brief positive, and then they were negative again, and very few symptoms, so hardly any symptoms. And then there were 7 people, so those are the purple ones at the bottom, that we call abortive infection. They had no positive tests and no symptoms. So despite getting the same viral titer basically inside their nose, they had no symptoms. So these lucky 7 individuals that we were able to study. And in this study design, basically, again, the special thing is, of course, because we know that the inoculation is at day 0 with the virus, we're able to also, for each volunteer, get the day -1 blood and nasal swab samples. So we have a sort of baseline, and of course that baseline is different for different people. And again, in a normal disease study design, you would have only the disease sample, not the healthy reference sample for that individual person. So having this longitudinal data from day -1, day +1, 3, 5, 7, and so on is incredibly precious, because it allows us to profile the progression basically of every single person longitudinally. And then we studied the nasal swab and blood data and analyzed it by single-cell RNA sequencing, TCR, BCR, and also multiplex antibodies, so 200 antibody panel for cell surface markers on the blood samples to give us these detailed immune profiles also at the protein level. And I should also point out that the challenge study itself was published a couple of years ago in Nature Medicine, and this is about the actual model. So because of course it took a while to basically grow the viral strains, optimize the titers, and so on and so forth. So what we analyzed was not only the blood, but also the nasopharynx. And what I'm showing here is immune cell populations sort of at day 1, 3, 5, 7, so on post-exposure, calculated as the abundance of those cells in terms of fold change over day -1, with a general linear mixed model. And you can see there are B cell populations, myeloid populations, T cell populations, and so on, and their kinetics kind of relative to day -1. And for the people with sustained infection, you can see that the immune cells are kind of first somewhat actually depleted because they're going into the blood. The monocytes are going into the blood out of the nose. And then from day 5 onwards, we can see kind of immune infiltration coming into the nose in response to the infection. On the other hand, basically, I mentioned those 3 people who only had 1 positive PCR test, so just a very, very transient infection, and they show already at day 1, so this is a very different pattern, massive infiltration of immune cells into the nasal epithelium. So this kind of can give us a hint as to explaining why those people clear the infection so quickly, is because they have this difference at day +1 relative to here, the people who have sustained infection. And then the third cohort are the ones who completely defend themselves to infection. And we can see there's actually not huge kind of changes in immune cell status in those cells. So what is it that protects those people from infection? So we did an unbiased differential expression analysis of all the cells in the nose, a nasopharyngeal swab on that side, and the blood in terms of calculating pre-challenge, so at day -1, what's the difference between the expression pattern in those people who completely defended themselves against the infection versus the people that had sustained infection? What's their baseline difference? And really the only clear gene that was statistically significantly kind of highly overexpressed in the people who defend themselves was this innate immunity cofactor HLA-DQA2. And so the innate immune system is of course the immune system in all of our cells that's defending us against the virus. This particular cofactor in the nose and in the blood that we found it is present in myeloid cells and B cells. So it's a non-polymorphic class 2 MHC molecule. It's been linked to milder COVID-19 progression in other studies, which also suggests a potential role in protection against infection. And so we don't know the detailed mechanism, but what we do know is that we see this particular expression pattern, and it's restricted to monocytes, macrophages, sort of myeloid cells, and also B cells. And so to summarize this COVID-19 story, basically we see distinct patterns in the early cellular response between different patient mini-cohorts, I'll say, that allows us to stratify the patients clinically, but then we also see the differences in their cells and expression patterns. And that pre-exposure immune signature that's associated with protection is this higher baseline HLA-DQA2 expression, this non-polymorphic MHC class 2 cofactor in myeloid cells and B cells. And so this kind of data and study with time inference basically we think can help in terms of vaccine studies and showing how different subtypes of people may respond, and in terms of also in informing us about how to respond and understand the immune system and its variability in different individuals for future epidemics and pandemics. And this paper, I'm not giving the detailed credits here. This was a collaboration with Marko Nikolić's group in London and a postdoc in my group, Rick Lindebaum, and a student in Marko's group, Kylie Warlock, led the collaboration between our groups and was just published, I think, a couple of months ago in Nature. So I want to move now onto more classical, I'll say, human cell atlas examples where we are studying healthy reference tissues, not specifically from the point of focusing on a specific disease, but in terms of our mapping of the healthy human reference body and gaining a deeper understanding of cell types, cell states. But what I also want to emphasize is from the atlas, so from the spatial localization of the cells relative to each other, we learn about how they're communicating with each other, which is telling us intrinsically about their function. And in particular, the reason that I also called this talk Mapping Molecules to Cells is that the tissue niches and the cell interactions and basically the way these communities of cells coordinate in their communication is revealing incredibly exciting and unexpected new insights into how our tissues and our organs function. And I'm going to start at the beginning of life with the placenta and then talk about a critical organ, the heart, and finally, if we get time, the thymus. So just going very quickly, the placenta was the first organ that we studied in terms of human single-cell genomics in my group. And the reason that we were interested in this was that we were coming from the point of view of the immune system. And of course, as you all know, basically the immune system has a role in detecting non-self, right? And basically killing cells that have non-self. So in terms of the SARS-CoV-2 virus, T cells basically and NK cells and so on kill all the cells that are infected with the virus in order to clear the infection. And so in the placenta, basically there's a kind of mystery in the sense that the trophoblast cells in the placenta have half of the alleles from the mother, half of the alleles from the father, and of course a lot of the paternal alleles are different from the maternal alleles, and so the maternal T cells, NK cells, etc., would be trained to recognize the paternal alleles as non-self. And yet, basically in this very special organ, which is crucial to pregnancy success and actually every single one of our lives, because it of course provides oxygen and nutrients to the embryo, basically the immune system is active in terms of protecting the barrier, the maternal-fetal barrier. It's also active in terms of helping to restructure the uterus, the decidua, and allow a change in vascularization that can deliver more oxygen to the placenta. But at the same time, basically there's a tolerogenic environment that's set up. And what are the fetal cells that are meeting the maternal immune system? Well, they're trophoblast cells that are coming and they're actually shedding kind of off or budding off the placenta and entering into the uterus, which in particular the endometrial layer, which is called the decidua in the pregnant state of the uterus, and they're invading into the different layers until they meet the myometrium and stop, basically, which is the outside muscular layer of the uterus, and then basically remodeling the vasculature, as I said, but also basically having a peaceful kind of a coexistence with the trophoblast cells rather than rejecting the placenta. So part of the mechanism that was known before is sort of hormonal mediated, although the mechanism was incompletely understood. Part of the mechanism that was known before, which is also conserved in mouse, is through chemokine exclusion of killer T cells. And part of the mechanism that was known was that NK cells have a specific state that's different from the natural killer cells in the rest of the body. But basically the detailed molecular mechanisms weren't known. And I want to also emphasize that because we're studying these cells at the sequence level, so we with Muzz Hanifa's group in Newcastle, we got ethical approval for also sampling maternal blood, and that allowed us to basically integrate the sequence from the blood and the fetal placenta and the decidua, which is in gray. We can then identify in an unbiased, sequence-driven way which are the maternal cells and which are the fetal cells. And that allowed us to identify macrophage states that are either decidual or fetal. And what that allowed us to show was that the macrophages that are patrolling the placenta and stuck to the outside of the placenta, which had been thought to be of fetal origin, are actually of maternal origin. So basically the cell atlas data provides new insights, in terms of understanding the origin of the cells. And it also, as I mentioned, allowed us to distinguish those special NK cells in terms of 3 different subsets that have different levels of activation. And it allowed us to trace precisely what are the cell-cell communications, basically what are the receptor-ligand interactions that are keeping these cells kind of peaceful and calm and not killing. So what is sort of keeping them in check? And how did we decipher those individual receptor-ligand interactions? You can see there's special HLA, HLA-G, LILRB interaction. And so this was known before, but it wasn't known exactly which trophoblast and which NK cell state is interacting, and that there are these 3 different NK cell states. And basically only one of them has that interaction. The others have other interactions, both with on the fetal side with the trophoblast cells, but also on the maternal side with stromal cells and macrophages that are kind of contributing to the tolerance. And where was this idea basically coming from to take the suspension cell data? Remember, single cell genomics is a priori with this technology, it's suspension cell, but we wanted to understand even from the suspension cell data, how are the cells fitting together in this puzzle, basically in this very dynamic tissue. And here, what my background in protein biophysics was useful. You can see here I gave a lecture a few years ago at the Nobel Symposium on Human 3D Proteome with Demis Hassabis and myself and others that were speaking in that public lecture session. And that's basically because a lot of our earlier work was on understanding protein complexes. And so this work which was using structural data, protein subunit stoichiometry to understand protein geometry and assembly, and we had this 3D complex database. What I basically developed with Rosa Vento-Tormo, who was the talented postdoc in the group, and Mirjana Efremova, was that we developed Cellphone as a database to take the receptor-ligand interactions that were known from 3D structures and have awareness of the subunits that are needed to form the correct functional complex, both on the ligand side and on the receptor side, and then predict possible cell-cell interactions. And in this early work, there was a question before about how did we sort of piece together these suspension cell data sets before we had spatial data, spatial genomics data. Well, what we used was prediction frameworks like Cellphone. We used integration with immunohistochemistry. We did single molecule fish experiments to then place these cells and understand where they sit in the complex tissue. And what we showed was that there were also a layered architecture of different stromal cells, different fibroblast cells in the placenta, and this wasn't really kind of described before in a molecular sense. So histologically, it was known that there were different densities in the different layers of the first trimester uterus, so this is early pregnancy, but it wasn't really understood at the molecular level what are the different cells that are defining those layers. We defined the epithelial compartment, as I've mentioned, different immune cell states, different trophoblasts, so the syncytial trophoblast, extravillous trophoblast, and so on. And this was really the first human organ, I would argue, that was mapped basically in this comprehensive way across all the different cell compartments, and also biopsied across all the different tissues within the placenta, and of course including the maternal side. And so what we learned was what are the niches that mediate the tolerogenic environment, basically, in this context, and move us forwards in understanding the tolerogenic environment and the remodeling of the tissue. So I'm going to go on to a second short story about another organ that is absolutely critical to sustaining life, and that's the heart. And of course, the heart pumps the blood around our body and delivers oxygen. And it's a really complex organ that's absolutely fascinating in the sense that it's not just a pump, a muscle, but it also has its own sort of brain, which is the cardiac conduction system that's electrically conducting signals from the sinoatrial node, which contains the pacemaker cells, to the two atria and the two chambers to basically contract in a harmonized way and deliver blood around the body. And so we had started collaborating with groups in the US and in Germany and in London on studying suspension cells in the heart. And then more recently, we worked with Michaela Noseda at Imperial College and Norbert Hübner in Germany to expand our study and to include the cardiac conduction system that you see in gray there, which is the sinoatrial node, where these spontaneously firing pacemaker cells are located that determine the rate at which our heart beats. And you can see the other locations of sampling of the tissues of the human heart there in terms of the two atria, basically at the top, and the two chambers, and then the two nodal regions. And what we found from the single cell and single nuclear RNA sequencing data was 12 cell types, so the cardiomyocytes, which are the muscular cells, the endothelial cells, the vessels, and then immune cells, lymphoid, myeloid, also stromal cells, the fibroblast, and neural cells. So there were 75 fine-grained cell states that we defined within the 750,000 or so cells. So the heart is as I said, the beating rate starts sort of in the let me just see if I can make this little video go. Yeah. So the beating rate is determined by the rate of firing of the pacemaker cells, which then conduct through the Purkinje fibers to the Purkinje cells, and then coordinate the contractions. So if the pacemaker cells don't function properly, basically there's massive challenges in regulation of heart rate, and also conditions like atrial fibrillation, which can be fatal. So if we map these cells and reveal their molecular identity and their content, we can gain deeper insights into heart function and potentially also learn how to engineer the cells in the dish and restore failing hearts. So the way we identified these pacemaker cells, and this was really heroic work, which was a collaboration with groups that helped us get these declined hearts from deceased transplant donors that were declined for transplantation, and then dissect them with James Cranley, who is a cardiology clinician scientist PhD student in the group, who's now a clinical lecturer with us, and collaborators in Newcastle and London. And during the pandemic, it was really hard to get any human tissue because transplant was basically shut down to a minimum. And so James and Kazumasa is a Japanese clinician scientist postdoctoral fellow in the group, worked together to basically travel around to London, to Newcastle in the pandemic, and initially only had 2 hearts. And they dissected them basically to get these different regions, and then tried to identify these very, very tiny nodal regions that contain the pacemaker cells, which are really hard to see surgically, because also this region is in slightly different positions in different people or in different organs. And so we actually worked with Professor Yen Ho, who was a cardiac anatomist at Imperial, and she helped us identify which were the likely nodal regions from histology, and that really helped us zoom in onto these regions. And in the initial 2 hearts, which I'm showing the data here, we are very lucky in catching about 50 cells that I'm showing in blue that have a slightly different transcriptomic pattern to the cells in orange that are working cardiomyocytes. And so we suspected that maybe those 50 cells that are kind of distinct from the working cardiomyocytes could be those pacemaker cells that are high in the calcium channel that you can see up there, CACNA1D, and you can see the working cardiomyocytes have this high sodium channel expression, SCN5A. And down here, you don't need to worry about the details, but what I'm showing is the expression patterns of ion channels and GPCR, sort of cell surface receptors, in neural cells and in working cardiomyocytes. And this box are the pacemaker cells, and you can see they're different and have a distinct channel and receptor expression pattern compared to the cardiomyocytes and neuronal and glial cells. And what gave us more confidence was also that we integrated this suspension cell data with spatial transcriptomics. So this was 50 micron Visium spatial transcriptomics. And with the histological annotation from Yen, we identified the nodal region versus the the myocardium, and the expression pattern of the pacemaker cells mapped to this inner nodal region. And what we can also find in the outer nodal region was patterns of fibroblasts, adipocytes, and so on. And the other interesting point that I want to make here very quickly is that in the spatial transcriptomics data, we can see differences between inner and outer nodal regions that are actually hard for the histopathologist to identify microscopically. So in that sense, the genomics data is kind of providing a new microscope or a new way of doing histology. And of course, what you can imagine is that there can be like a feedback with machine learning between informing us about the patterns from the histology and the molecular genomic signatures to now define these tissue niches in a new and unbiased way at higher resolution. And if we kind of show schematically, basically what we are kind of finding here in terms of the niche is that the pacemaker cells have special glial cells, which are these yellow cells that we confirmed by microscopy to kind of give us confidence that yes, indeed, this is what we are seeing in the genomics data, our pacemaker cells. The glial cells are kind of hugging them and nurturing them and interacting with the autonomic nervous system that's sending the signals from our brainstem basically through the sympathetic and parasympathetic neurons into the pacemaker cells. So although the pacemaker cells are spontaneously firing, so they start firing from the very beginning of embryonic development as the heart develops up to the end of your life when there's the last breath and the last firing. They keep on spontaneously firing, but the rate at which they fire is modulated by the neuronal input through the parasympathetic nervous system that's decreasing, the sympathetic nervous system that can increase the rate, and also the endocrine system, so hormonal input. And then we saw special fibroblasts that are providing an extracellular matrix, kind of that these cells are structured in. And of course, there needs to be conduction to the Purkinje fiber and the atrioventricular node, but also insulation, so not in a random kind of conduction. So what we thought was, given this data for the heart, and given the fact that there are a lot of drugs that are modulating heart rate, so-called chronotropic drugs, can we basically take the database of approved drugs, so this is the openly available Chembl database that has approved drugs, and what are their molecular targets? Sometimes there are multiple targets for one drug, and this database, Chembl, also contains 2 compounds, and can we match it against the cell types that we have in the heart, and including the cell surface receptors and channels. And this is the drug-to-cell computational sort of framework that we make available on GitHub. So one kind of really surprising finding that I want to highlight is that in these human pacemaker cells, but not in mouse, we see expression of the GLP-1 receptor. And the GLP-1 receptor is the receptor for GLP-1, which is kind of metabolized into glucagon, which is, of course, the hormone that controls how hungry we feel. And the GLP-1 receptor analogs, basically, sort of GLP-1 analogs, GLP-1 receptor agonists like semaglutide and liraglutide are used for treating diabetes and obesity, marketed as Wegovy and Ozempic and Mounjaro and so on. And what was really surprising was that we find that the pacemaker cells have receptors for those analogs. And what this suggests is that the clinically observed 6 beats per minute increase in heart rate right at the beginning when people are put on these drugs, that's reported in clinical trials, could be operating via a direct mechanism of action of the drug on the pacemaker cells. And this was unexpected because, of course, the GLP-1 is binding to hypothalamic regions in the brain and suppressing appetite, so people don't feel that they want to eat more. And so the expectation would be that this would be via neuronal mechanism. But here, with the human cell atlas data, we're showing that this could actually be a direct mechanism of modulation of heart rate via the endocrine system. And so that's really interesting because what it illustrates is that the human cell atlas basically is giving us information on drug side effect mechanisms and kind of We have validated this mechanism both in vitro with iPS-derived cardiomyocytes, and recently there was a publication from a cardiology group that works on the pig model that this is actually conserved in the porcine large animal model. So again, it's an illustration that the Human Cell Atlas can act as a molecular guidebook. So the first part I mentioned SARS-CoV-2 entry factors, and that we can use the Human Cell Atlas to map basically where the virus is entering. This is showing that we can also use the Human Cell Atlas to map where drugs are acting and where hormones are acting. So we have cell-cell communication in terms of niches, but also in terms of long-distance signaling. And of course, with drug to cell, we can match drugs to cells and cells to drugs and use this in potential applications for understanding toxicology, side effects, and also repurposing. And so what we defined here was not only the individual cells, but also niches for cardiac conduction. And the important point I also want to make in this section is that we can define with cell phone, I showed in the placenta, is paracrine signaling, so that's close by signaling between cells and niches. But here also endocrine signaling, which is the hormone system that's coordinating the organs in our body and is traveling over long distances, over centimeters, and even meter-long distances by these small signaling. I want to finish with a last story about the immune system, the thymus in particular, which is where our T cells mature. Now, this is a little organ. We're now traveling a little bit sideways because it's a small organ that's actually stuck to the heart. It was only discovered in the 1960s as an immunological organ. Before that, it was just thought to be a little piece of fat that stuck to the heart. And this was described as the site of T cell development by Jacques Miller and colleagues. And what happens is that hematopoietic stem cells enter, and then the T cells mature in the organ. And the story that I'm going to tell you about just came out last week in Nature as part of a Human Cells publication bundle that Partha and many, many others contributed to, where we had over 40 papers that were released together from the community. We had released a lot of them online before in bioRxiv or in advanced online publication, but they were in the journals. They were all published together in Nature Publishing Group in different journals containing themes of data integration. So we're now in a phase in the Human Cell Atlas community where we're integrating published data from many, many groups to make reference objects for organs like the lung, the brain, the eye, and all of the 18 biological networks in the Human Cell Atlas. And so there are a lot of papers in this bundle that are examples of that, also focusing on human development, so embryonic and fetal stages, and then finally publishing computational and also experimental methods that are useful for basically advancing the project. And so this data has just been published, so again, I'll go fairly quickly, just like through the other stories, to just say that the thymus is where hematopoietic stem cells enter, the T cells mature, and then exit to colonize the tissues in the body. And of course, T cells are also interesting from a therapeutics point of view because they're used as drugs themselves as products for tumors and liquid tumors, and also more recently autoinflammatory diseases like lupus. And the question that we were asking here is, so we talk about the Human Cell Atlas, and I've told you about 2 organs where we map multiple tissues, right? The placenta, we were sampling from Deciduo basalis, Deciduo paritalis, all the different regions of the placenta itself. And the heart, basically the nodes and the myocardium from the different chambers, the apex and the septum and so on. But in none of those projects did we map an entire organ within one reference framework. Now, that's obviously hard because human organs are so large, so how can we generate a single model? And how can we also model multiple different modalities together? So imaging, spatial genomics, single cell, single nuclear transcriptomic data, and so on and so forth. So the thymus is a pretty small organ, basically a few centimeters in diameter, and the individual lobes can be on the order of less than a centimeter. So it's a kind of repeated structure of the organ where we have outside layers that are the cortex, and inside in the center is the so-called medulla. And so the idea that Nadav Yayan, who was a postdoc in the group, came up with was to model basically all of the data sets, whether they are suspension, spatial, imaging, and so on, onto a single nonlinear axis, where we have points of reference, landmarks, a few landmarks, basically the outside edge, the corticomedullary junction, then the center, and we basically have a -1 to +1 axis that we project all of the different data sets on with nonlinear relationships between the landmarks. And so this, you can sort of think of this as mapping each lobe of the thymus onto like the different layers of an onion, and what that allowed us to do was to then basically gain very high resolution, detailed insights into where in this model of a thymic lobe, where are different cells positioned relative to each other from the outside into the center, both epithelial cells, immune cells, fibroblasts, vascular cells, and so on. And also how are there gradients of expression of signals like cytokines and chemokines that are basically like a vector field of input into the developing T cells as they're maturing. And basically the sort of one of the things that Umesh asked in the previous talk was, can you model the developmental trajectory of cells from progenitors? And this is one example where the T cells are basically maturing also spatially as they travel from the outside to the inside. So the hematopoietic stem cells enter near the corticomedullary junction, and the T cells mature as they go through. And this sort of mathematical kind of modeling framework has basically helped us to get a very detailed atlas of the entire organ. And this organ axis software is on GitHub and can be also applied to many other tissues. So we're using it in the lab to model also cardiac, muscular wall. You know, you can project this basically onto any organ that has landmarks and gradients, which is many, many tissues and organs in our body. So I want to finish by actually just talking a little bit about applications of having this kind of detailed map with all the different tissue niches basically defined for the purpose of engineering cells. And so for the thymus, we have this thymus cell atlas, and where we have the detailed information about how T cells are maturing. And I've just, I'm not talking about this in terms of the individual cells and molecules, but just at a high level, and you can read the details in the paper that just came out last week. And so this data on the individual transcriptional regulatory modules, the individual transcription factors, and the inputs basically in each niche, the sort of signaling input, can inform us when we make iPS-derived T cells. And so a very talented clinical lecturer now, who was formerly a PhD student in my group, Chen-Chu Suo, has basically implemented the iPS-derived T-cell maturation protocol, the so-called artificial thymic organoid from Gay Crooks's lab at UCL. And what she then can do, of course, is to basically modify this protocol in terms of engineering the iPS cells with different transcription factors that are need to be upregulated based on the in vivo thymic data, and also add different factors into the dish during the protocol, basically, that we have in cell culture. And so that's kind of the current status of using the in vivo human cell atlas data for better engineering of cells in the dish and sort of precision design of protocols for making iPS-derived cells and for making organoids. So taking stem cells or primary cells that are derived from the body and then directing them to make particular cell types using the right conditions and the right molecular ingredients is where the human cell atlas can act as a blueprint, basically, for these engineering applications. So the human cell atlas, I've talked about the human cell atlas as a guidebook, basically for where molecules are in the body, but we can also think about it as a blueprint, basically, for engineering cells in the dish. And of course, those engineered cells can be used to understand hidden processes involved in human cell and tissue development, so doing mechanistic assays that we can't do in the human body, to model and better understand diseases, screen drugs, and ultimately, basically, of course, as therapies, which is already for CAR T cells happening. But in the long term, also for transplantation, like clinical trials are now ongoing for iPS-derived dopaminergic neuron transplantation in Parkinson's disease patients, in a clinical trial by Bayer that's going on in New York, in Sweden, and in Cambridge. And those kinds of applications, we hope that the human cell atlas data can help to engineer cells more faithfully to mimic their in vivo counterpart. So I want to end by taking a short look into the future and dreaming about the therapeutic potential of engineered cells and tissues, which we all know is enormous. But currently, that process of making those cells is really expensive, laborious, time-consuming, and imperfect. And so what I hope is that in the future, we'll be able to make synthetic cells and program them in whichever way we want. So how could we do this most efficiently? Maybe synthetic genomics could help us. So at the moment, synthetic genomics, chemical synthesis of chromosomes, has been achieved for whole yeast chromosomes, which are obviously much shorter than mammalian chromosomes. But maybe in the future, these methods could be scaled up to synthesize entire mammalian chromosomes. In particular, using the human cell atlas data, we could design chromosomes that only have the genes that are needed for a cell type and the regulatory elements that we define from single-cell attack sequencing. And so we could determine the desired behavior of cells to either mimic in vivo cells or make cells with completely new properties. So we would have a highly controlled means of making cells. And how do we figure out which genes to use? Of course, we use the human cell atlas as a blueprint. So I want to finish there by saying that the human cell atlas is already revolutionizing our understanding of the human body, even though we're far from completion. The human cell atlas can be used as a framework or a blueprint to help restore human health in many ways, and also as a guidebook to tell us where each gene is expressed at the level of individual cells, cell types, and cell states. So the basic knowledge that we are all building together in this large interdisciplinary human cell atlas community that's globally has over 3,600 members will have a huge biomedical impact in the future for both research and for medicine. And I'll stop there, say thank you, and take any questions.
Thank you, Sarah, for a very brilliant talk and introducing us to exciting works and possibilities with the human cell atlas. We have time for questions, comments.
Thanks for a very impressive talk. I have two questions. One is, do we know the molecular formula of a cell?
I mean, we're getting there in some sense.
Close to that.
Yeah.
The second question is, like towards the end, what you're promising, synthetic cell and synthetic genomes, they don't have life. How would it affect the treatment?
I mean, I'm not promising, I'm dreaming about what could be possible.
The fact that they don't have life, would it matter, the synthetic cell and synthetic genomic?
I mean, it depends on what you mean by life. If life is the ability to replicate, they could replicate. You could design them to enter the cell cycle and proliferate. So in that sense, cells are alive in the dish. I mean, depends on what you mean.
Yeah, simply wonderful, fantastic. A huge body of work. I think you condensed it in an hour's lecture. So my question is, are we? I'm sure it can do many things. Yeah. Are we ready in a clinical setting to provide molecular and cellular explanations to say what we already have, like ultrasound images or MRI, and therefore subtype patients and give the recommendation for treatment?
So I think your question is super relevant, and I had an extra slide basically here. I think what we need to do is ultimately link the cell atlas data in suspension cell, which is 0-dimensional, 2-dimensional, to larger volume imaging technology. So what I'm showing here is hierarchical phase contrast tomography, but there's also medical imaging technologies like MRI. And this is really this sort of multi-scale mapping of the human body is something where we have a program funded by the Canadian Institute for Advanced Research that funds 2 little workshops per year with about 20 people, where we're thinking about how this might be possible. And in fact, at the moment, there's a nuclear medicine clinician who's spending a sabbatical year in my lab, who's sort of thinking about what he calls the imageable genome, so using PET, MRI, the tracer technologies that you have in medicine. Can we integrate them with the molecular data from Human Cell Atlas and then help to interpret the medical imaging technologies? So yeah, it's a very relevant question.
Yeah. Hello, Sarah. So very interesting talk. I have 2 questions on the SARS-CoV-2 infection. First of all, congratulations for getting the volunteers. And also, I know it's hard, but with this, nearly 40% are non-symptomatic. So can it be generalized? That's the first part. And the second one is on the organ. So for instance, placenta. So can preeclampsia be understood better? Or if there is a heart failure, can it be understood better using this single cell? Are we ready now?
Yeah. So great question. So on the question, the first question about the extrapolating, I mean, we would, basically that is the prediction, is that there are people who completely defend themselves from infection. Whether they're those people who are roaming around who have the virus in their nose but have no symptoms, could still transmit. And that's obviously kind of, important to know, and it's a threat. So we would predict that it would be, some significant fraction, remembering that these are 20 to 30-year-olds. So of course, what we know is that with older ages, there's more severe symptoms, and actually in children, they're the most resistant. So this data is specifically for that kind of age group. And we've also studied kids and their response to COVID-19. And that's basically very interesting from a mechanistic point of view in terms of their more active innate immune response. Yeah. So the second question was about how the healthy reference map of organs like the placenta and heart and so on can help us understand disease, and sort of preeclampsia as one example. And basically, that's what we are using this data for, to some extent in our work, is to map, for instance, genetic variants associated with preeclampsia, also to do with, not on the placental side, but on the endometrial side, to do with endometriosis and so on. And what we hope is that we can gain more mechanistic insights to advance therapeutic approaches for those.
Yeah. Before we take the next question, I request Sumesh to check if there are any online questions typed in, please. Yeah. Yeah. Can we have your question now, please?
Yeah, very nice talk, madam. With reference to your first half of the talk with SARS-CoV virus, was there any receptor difference or receptor molecules with abortive group to the transformants or the infective ones? B080 or receptors which are responsible for internalizing the DNA of virus.
So we didn't see any differences in expression of the receptor factor, the factors. I mean, this was really an unbiased differential expression analysis, and the only gene that popped up as clearly significant was that HLA-DQ2, so it was MHC class 2 cofactor. Yeah, no receptor differences.
Thank you.
Yeah.
Sarah, when we talk about resistance to an infection, is there not a quantitative element associated with it in terms of the dosage of infection?
Yeah.
So were all of these 16 people at the same dosage
Yes.
level?
Yes.
So what I'm thinking is these people who are mildly resistant or completely resistant, as you call them, if we
Yeah.
increase the dose,
Yes.
could they not transform into being?
Yes. It's dosage dependent. You're right. So the dosage is optimized in these trials to achieve some, substantial subset of the cohort that gets infected. But it is definitely dosage dependent.
Thank you for the talk. Just a couple of questions. You mentioned that the seronegative challenge. How much time did it take to actually, it's the public emergency sort of scenario?
Yeah.
How quickly could you finish all this and put data out for it to, inform others?
So for the for the human cells community to study the healthy data was super fast. That was like 6 weeks. From when we started in February 2020, I think in March we posted the data on an archive. bioRxiv didn't take it, we posted it on archive. So that was super quick. Doing the human challenge study was a lot of work. So even though it was an emergency, because of all these optimizations, like what's to actually culture the virus first, optimize the titer and so on. This is a huge collaborative effort, and the challenge was already published 2 years ago. It was just we had a lag also in doing all the genomics, processing the data, and so on. So that's why we've only published now. And what I should also say is, obviously, this is at this point we are all resistant because we've either been vaccinated or infected, basically, at least in the UK. It's so endemic now, SARS-CoV-2, that now everybody is seropositive. And so what's been started now is a seropositive challenge study in Oxford. And there the interesting thing is actually with this titer, nobody, with the original strain, nobody gets infected. So basically what they had to do was take other strains and massively increase the viral titer. And only then do you see any infection in seropositive patients. So vaccination and immune memory, like from infection, it really works. It really works.
Firstly, wonderful session, ma'am. It was very inspiring, especially for us undergraduate students who are attending, and we are learning a lot about biomedical. Firstly, my question is about the HLA-DQA2 gene.
Yeah.
It's kind of a personal question because back in 2021, my entire family had gotten COVID, but I had gotten the least amount of symptoms, or at least almost nearly no symptoms. I was always curious as to why that was. So is there any evidence as to its expression? Is it affected by age or the expression of the HLA-DQA2 gene? And about heredity, is it that certain populations might have more, might show more expression of that gene, or
It's a really good
certain
question.
individuals?
And I'm ashamed to say that we haven't studied the age dependence, but
Okay.
it's a great idea. What is known is that there are also other mechanisms. So in kids, like in younger people, we've shown a couple of years ago in a paper that not only HLA-DQA2, but actually the whole innate immune response, interferon, TNF, and so on, is much higher. So those interferon type 1 and TNF modules are expressed in all the cells in our body. And in kids, they're much more highly expressed. So pediatric, like from age 1 to 16 and so on, they're much more highly expressed. And so I don't know if you would have fallen to that age group, like more highly expressed than adults. What Mala Maini and collaborators from UCL have shown is that there's also some protection that's conferred by memory T cells where there's cross-reactivity between the internal antigens of circulating seasonal coronaviruses and SARS-CoV-2. So that could be another mechanism. If you happen to have memory against a circulating coronavirus that has conserved internal antigens, then that can also give protection. So this is not the only mechanism. There's also pediatric kind of mechanisms that we've shown, and Mala Maini has shown evidence of these memory T cells from her healthcare worker cohort study.
Thank you, ma'am.
Thank you. While you transfer the mic to the next person, there's one question online by someone called Madana Navalu. The question is, do cardiomyocytes divide? And what type of glial cell is there in heart?
Yeah, so that's a good question. So cardiomyocytes in adult human, are thought to be terminally differentiated. And in our data, we don't see any evidence for any cell proliferation whatsoever. So it's different from mouse or zebrafish. In human, yeah, at least in adult human. Of course, in developmental data from the heart, there is proliferation. The glial cells, yeah, it's a good question. The glial cells are, the glial cells are obviously specific to this niche. I mean, in terms of how similar they are to glial cells in other tissues in the body or to the brain, the different glial subsets, I can't comment on that right now, but the data will allow you to look at markers and answer that question.
Thank you.
Yeah.
We will have one last question from here.
Thank you very much for such an interesting talk, ma'am. Wondering that we now know from the single cell RNA or spatial the overall transcriptomic profiling of the gene expression of individuals.
Can you hold the mic up so all of us can hear you?
So I just wanted to ask whether the transcriptional factors, have you tried looking into the transcription factors that are regulating the different cell types, the mapping of different transcription factors in each cell type across different conditions? And secondly, when you spoke about site sequencing, we know that there are certain limitations about say about 200.
You really need to hold it closer to your mouth, please.
Yeah, so we have looked at the transcription factors in different subsets. And in terms of cardiomyocytes, we are trying to engineer iPS-derived cardiomyocytes towards the pacemaker lineage through addition of transcription factors that are specifically skewing them towards that lineage. And also actually in the artificial thymic organoids, we're also using lentiviral delivery of transcription factors and other delivery methods for transcription factor expression into the iPS cells.
And also regarding the site sequencing
Yeah.
that you have mentioned, that we have a certain limitations of probing about say 200 proteins or
Yeah.
so. So what can be the sensitivity? How can the sensitivity be increased for say?
I mean, there are expansions of the antibody panels now, and there's also intracellular site-seq now. So there are both in companies and in academia, there's sort of development and optimization of the oligo-labeled antibody panels to expand the sort of proteomics data at the single cell level.
Okay,
Yeah.
thank you. Thank you all of you for being here with us, and
Thank you.
we thank Professor Teichmann for a very brilliant presentation of a really nice work, exposing us to lots of exciting things in this field. On behalf of the Indian Academy of Sciences, we wish to thank you with some mementos.